Dear professors, colleagues, students, we're glad to welcome you at our third issue of Web Library of Neuropsychiatry, managed by International Center for Education and Research in Neuropsychiatry, Samara State Medical University. And last week of May 2021 is nominated as Schizophrenia Awareness Week, and also 24th May 2021 is the World Schizophrenia Day, and we're happy to support this direction and to spread the word on behalf of our center about schizophrenia. And today we have a prominent speaker, Professor Paul Cumming, from the University of Bern, Switzerland. And Professor Paul Cumming is the expert in neuroimaging, and the lecture will be devoted to molecular imaging of schizophrenia. The floor is yours, Professor Paul Cumming.
Thank you for that introduction. I have chosen for today's talk to focus on molecular imaging of schizophrenia, as opposed to the more general topic of neuroimaging in the broader sense, which often includes magnetic resonance imaging. I believe that that topic is something for another day. So today, I will instead talk about some typical findings obtained by positron-emitting tomography and its allied technology of SPECT, revealing the molecular basis of schizophrenia. Now, of course, I don't need to explain much about the clinical diagnosis of schizophrenia to this audience, but let us say that this has been among us for centuries, if not millennia, as far back as we can say. Here depicted in a famous illustration of the Bedlam Hospital, as it came to be known, St. Mary of Bethlehem Hospital, formerly in London, since the Middle Ages, a place for concentrating the people who would now be called mentally ill for whatever causes, organic or psychological. Now, this place was notorious and has become a term of, in the English language, Bedlam as being a place of general disarray. And as you can see, it was until fairly recently, until the late, I think, 19th century, it was possible for people to pay a ticket to visit Bedlam. Here we see two elegant ladies who are viewing the site as kind of a form of entertainment, which reflects how attitudes have changed. And indeed, that is something in the matter of a product of the Renaissance. Here, a famous painting showing Dr. Pinel, who famously unloosed the chains that were used to bind patients in a long-term psychiatric hospital in Paris. And he was viewed, and is to this day viewed, as the kind of initiator of the modern humane perspective on the treatment of the insane, now as they were then called. And of course, from that optimistic point of departure around 1820, we unfortunately had some setbacks in the course of attitudes towards the treatment of specifically schizophrenia, most notoriously in the inappropriate and really criminal use of the leucotomy that was popular in the mid-20th century, especially in the United States. Now, shortly after that photograph was taken in the 1950s, there was the advent of modern psychopharmacology for schizophrenia, with the discovery, I think, of chlorpromazine, or one of the very early antipsychotic medications, which has led to a vast diversification of drugs that have some degree of efficacy, at least against the positive symptoms of schizophrenia. And this pharmacology, their pharmacology, is presumed to give some hint about the underlying pathology of schizophrenia. There's a great plethora of possible variants of the original antipsychotic drugs. So I say that they come in many colors, but more specifically, they come in many chemical compositions. And this famous diagram, I think from the late Phil Seaman from Toronto, shows the observation that supported the so-called dopamine hypothesis of schizophrenia, The observation being that on the x-axis, the clinical potency for controlling schizophrenia from the very potent butyrophenones to less potent compounds such as chlorpromazine, which require gram doses per day, there is a clear association with the in vitro findings of the affinity of those very same medications for the dopamine receptor, as labeled with haloperidol. And so this, of course, does not prove causality, but it shows that the more potent the compound for treating schizophrenia and the lower dose consequently required, the more effectively it interacts and tightly it binds to dopamine receptors, suggesting that dopamine receptor affinity is the determinant of clinical efficacy. However, I want to be cautious about this and refer to the term syllogism, which is a logical term for a form of reasoning where a conclusion is drawn from about a missing middle connection between two things, such that all dogs are animals, all animals are quadrupeds, therefore all dogs have 4 legs, except those that have lost one in a tragic accident. So somehow this kind of logical thinking or fallacy prevails in the context of schizophrenia, like so. Antipsychotic medicines relieve the positive schizophrenia symptoms, that's clear, at least in many cases. Second, antipsychotic medications block dopamine receptors. Therefore, schizophrenia patients have too much dopamine or too much activation of receptors. It doesn't actually follow. It could be that the problem lies elsewhere, and it just so happens that dopamine blocking alleviates the symptoms. That's something that I think we all have to bear in mind. Be that as it may, a new era of brain research began with the advent of molecular imaging by positron-emitting tomography, which we call PET for short. And in this procedure, a pharmaceutical can be tagged, incorporating into its chemical structure a radionuclide that emits radiation in the form of a positron, which is an antimatter electron. And therefore, you can follow where the drug goes in the body by monitoring the emissions from the drug. But first, to do this, you have to make the positron-emitting radionuclide. And this requires an instrument called a cyclotron, which consists of 2 nesting magnets surrounding a vacuum space, around which particles are accelerated by alternating current to very high energy, in this case, a proton, which when bombarded against a stable isotope of oxygen, oxygen 18, yields by neutron absorption, by a chemical nuclear reaction, ultimately yields fluorine 18, which is a very unstable radionuclide that behaves just like fluorine. So this is actual transmutation of matter from oxygen to fluorine, yielding a highly radioactive product that decays with a half-life of 2 hours. So if you make it in the morning in the cyclotron, it's mostly gone by the end of the day. This is the inner workings of a medical cyclotron, which measures about 1.5 meters in diameter. And here you see the various targets where the oxygen could be placed to be bombarded from a beam extracted from the cyclotron. It's shielded. You don't want to be in the same room as a cyclotron when it's operating because it's fiercely radioactive. And the basic principle is that fluorine 18 decays to oxygen 18, returning back to from what it came from, with the release of an E+, that is an antimatter electron or a positron electron. Now, if that occurs in the medium of the brain, it doesn't have to travel very far before it encounters an electron somewhere in the water or any element of the brain matter. And opposite charges attract, of course. They interact because there is no force to keep them apart, and they annihilate, releasing all the energy that was bound up in that electron-positron pair, expressed as E equals mc squared, the Einstein formula, as 2 photons. It looks like a burst of light here, but more correctly, it's 2 photons going in opposite directions. One gamma photon, high energy gamma, going this way, and the other going that way, spread at 180 degrees. And they contain all the energy that was bound up in the matter of the positron-electron pair. So they're very energetic. They can go through, these gamma rays can go through a centimeter of lead. But they can be stopped with detectors. And here we see, I think, the first PET scanner built in the 60s, which had a ring of about 10 detectors that were placed like a crown over the head and wired together in such a way that the simultaneous emission of two photons, gamma photons, from an annihilation event somewhere in the head, barreling out away from each other at the speed of light, they encounter on one side of the scanner a detector, and another side of the scanner another detector, as shown here. And that event, if it happens in the same moment, counts as a decay. And by counting millions of those events, you can build a picture showing the distribution of radioactivity in the brain of the living person sitting here awake, more or less comfortable in the scanner. Modern scanners are much cleaner. They hide all the wires, and furthermore, they cost millions of euros, and they contain thousands of detectors. The more detectors you have, the better detail you can see in the image. So now, from the modern medical technology, I go back 200 years to the time of Pinel, in 1817 with the publication of the famous essay on the shaking palsy, now named after its author, Parkinson's Disease, who classically described it as involuntary tremulous motion with lessened muscular power, in parts not in action, therefore at rest, and even when supported, with the propensity to bend the trunk forward and to proceed from walking to a running pace destination, the senses and intellects being uninjured. Except for that latter comment, we now understand that Parkinson's brings with it cognitive changes. Except for that latter comment, this is a perfect clinical description of Parkinson's disease. So why am I talking about Parkinson's disease in the lecture on schizophrenia? Well, because of the central connection with dopamine. Here, a clinical picture, a paper showing the progression of micrographia, which is the lessening amplitude of handwriting and motion in general in a Chinese speaker who proceeded from a normal condition to a more compressed form of writing as her Parkinson's disease rigidity advanced. And in concert with that, her brain was undergoing a progressive loss of its essential neurotransmitter dopamine. As was first shown in the very early 1960s, originally based on autopsy observations here of the mesencephalon on the right-hand side from a normal individual, showing the highly pigmented dopamine neurons of the mesencephalon, and here a patient who died with very advanced Parkinson's, suffering a nearly complete loss of brain dopamine. And the association with schizophrenia and Parkinson's as opposite ends in some sense of a spectrum of too much dopamine versus too little dopamine was established early on by Professor Arvid Carlsson of Sweden, who discovered dopamine in the brain in 1958, I think. Someone had to discover it, and he went on to connect it with the control of movement and hypothetically with the chemical pathology of schizophrenia. Here in his famous 1960 experiment, he treated rabbits with the toxic alkaloid reserpine, depleting dopamine in the brain and rendering them essentially immobile. But upon giving them the precursor of dopamine, levodopa didn't exist then, but the racemic mixture, DL-DOPA, the bunnies perked up immediately and started hopping around like normal, thus restoring them, curing, in effect, their chemically induced Parkinson's disease. And at the opposite end of that spectrum, too much dopamine could also be a bad thing. Now, Arvid Carlsson, who won shared the Nobel Prize in the year 2000 for his discovery of dopamine, presented at his Nobel lecture this old figure, which I love because it shows in very schematic terms what a dopamine synapse consists of: a presynaptic structure containing mitochondria with monoamine oxidase in it, containing synaptic granules that store the dopamine and release it, and uptake sites that take it back up into the neuron, and enzymes for synthesizing dopamine from its amino acid precursor. The whole point being that the dopamine is supposed to act postsynaptically at receptors expressed on the target cells in the nervous system. And Professor Carlson, although we only met it once, I considered my mentor, and in fact, this is my shoulder. So I was taller than him, but in other respects, not to be compared. Now, with the discovery of DOPA and the synthesis of dopamine from its precursor amino acid, actually a Canadian scientist in the late 70s proposed the idea of labeling DOPA, dihydroxyphenylethylamine, not ethylamine, it's the amino acid, excuse me. Now, this is the amino acid containing the carboxyl group and the amine, now modified by the addition into the structure of fluorine 18, where there was normally just an ordinary phenolic proton. And that makes it radioactive, but doesn't otherwise change its chemical properties. So it behaves just like, almost like natural levodopa, and it is radioactive due to the fluorine 18. So they generate the fluorine 18 in the cyclotron in the morning, incorporate it into the chemical molecule of DOPA to make fluorodopa, and then use that as a PET tracer for positron emission tomography. And like natural DOPA, fluorodopa crosses the blood-brain barrier after intravenous injection. And when it meets a dopamine fiber, it is trapped inside that fiber because it's chemically converted by decarboxylation by the enzyme DOPA decarboxylase that makes the corresponding amine, which is fluorodopamine, still radioactive and now trapped inside the vesicles of the dopamine fibers. So that is the principle. By introducing a radioactive tracer, fluorodopa, into this pathway, we can label the living neurons by virtue of their ability to take up the tracer, decarboxylate it, and trap it inside granules or vesicles. And here, this scratchy old photograph is one of the first depictions of, some of the first depictions of PET scans with fluorodopa that were obtained when I was a graduate student at the University of British Columbia. Using fluorodopa, we see that a normal individual has high uptake in the dopamine-rich caudate and putamen, whereas people with Parkinson's disease and certain other syndromes have a very substantial loss of that dopamine capacity, especially in the putamen, and a relative sparing in the caudate. And that matches perfectly with autopsy results. So much for Parkinson's disease. There's no doubt you can look, actually, and this is an example of a radiological use of FDOPA-PET, because you could, if you didn't know already, diagnose Parkinson's disease pretty clearly by just looking at the scan. Now, the other side of the coin, a functional excess of dopamine in patients with psychosis was hypothesized since the 1960s, as first introduced by Arvid Carlson. And 30 years ago or so in Montreal, the first test of this was conducted using fluorodopa in a small series of patients with schizophrenia in the PET and psychiatry group, which then operating at the Montreal Neurological Institute. And the results are summarized here, where we show the average fluorodopa uptake in a series of 13 normal subjects with a high uptake in the caudate and putamen. This is the average of 13 scans, so each brain is kind of stacked upon each other and normalized. And the main finding is that the patients with schizophrenia, of which there were only 5, show a highly significant increase in dopamine trapping. There were only 5 because it was very difficult to recruit 5 patients with schizophrenia who were not medicated, because medication would interfere with the results. That was the initial finding, showing that there was, as expected, an increase in some sense in the dopamine level in the brain. We also scanned a few patients with complex partial seizures with interictal psychosis. an equal number of 5 cases. And you notice that they had an increase, but it was only on the right side, which happened to be the side where the epilepsy was in the dominant hemisphere. More about that later. So, but in a sense, increased fluorodopa signal is not, while it's present in schizophrenia, is not uniquely characteristic of schizophrenia. There are other conditions where that can happen. Because of that intriguing result long ago, many people tried replications, and Professor Oliver Howes in London and Kshitish Kapoor now in Melbourne compiled all the available studies up to about 5 or 10 years ago, and in a sense combined them into one single study of many subjects. This is called a meta-analysis. And the results of their meta-analysis are shown nicely here. In historical sequence, the first such study, the Montreal study from 1994, showed a very big increase in fluorodopa in a small sample of schizophrenia patients compared to controls. And then there were various replications, mostly, but not always, showing an increase, continuing to the 2010. And this actually is an old paper now, so there would be several more replications since then. And this trapezoid here represents the composite effect of all of the subjects who represent nearly 200 patients and 200 controls. Not something you could do very easily at one particular site, but by combining all of these different data sets together, it was possible to show that there was indeed an overall effect that was highly significant, p less than 0.001, which is once in 1,000. And that convinces me. One single PET study, like here, Reith et al., with 5 subjects is interesting, but not convincing. But if it holds true for several hundred patients, it emerges as being a real biological result. However, statistics is very harsh. In this figure, we see normal distributions, which would describe most PET results in a population. In the lower figure, this is what we would consider a good or diagnostic separation, where, for example, this might be schizophrenia, fluorodopa results, and this might be healthy controls. And if we were so lucky that we could scan somebody and just say from looking at it that they belonged with 95% certainty to one category, that would be diagnostic. However, for the case of fluorodopa-PET, although the averages are different, the distributions overlap very significantly. So we can say that the meta-analysis shows that there's something wrong with dopamine in schizophrenia as a population, but there are many individuals with schizophrenia who have normal dopamine results. I think this is a very important concept. So now we proceed to another representative study using a similar compound, carbon-11 DOPA, which is another PET tracer. And they had what is typical for a PET study group, 12 patients and 10 healthy volunteers. And you can see once again, as generally found, that Patel et al. In Karolinska were able to show that schizophrenia patients had an increase in fluorodopa trapping, or carbon-11 DOPA trapping. Interestingly, they found an asymmetry. If you recall, we also saw an asymmetry in the very first study, but in partial complex seizures, complex partial seizure patients, suggesting that asymmetry of the basal ganglia abnormality can be a characteristic of schizophrenia or psychosis from whatever cause. Not everyone sees it, but it happens often enough that I think it's a general phenomenon that there may be inherently an asymmetry of the dopamine system in schizophrenia. And this harkens back to 1960s vintage thinking by Crow in the 70s, and in Edmonton, my hometown in Canada, there was a psychiatrist who saw the link between epilepsy and schizophrenia, who also claimed it was Pierre Floor Henry, who showed that there was some association between temporal lobe epilepsy and dopamine asymmetry, linking perhaps the syndromes of epilepsy and schizophrenia. Now, suddenly I take a leap away from the dopamine system to metabolic PET studies with fluorodeoxyglucose, or FDG, which is a glucose analog that is widely used the world over for PET imaging, usually diagnostics, because it's highly taken up in tumors. For example, in these axial lymphoma lesions and throughout the groin area and in the lymphatic system in general here. Now, this patient was scanned with FDG at baseline and then was treated by chemotherapy and enjoyed near a complete cure, basically, of the disease, characterized by an absence of fluid FDG avid lesions. But you see, the doctors weren't interested in his brain. They were only interested in lymphoma sites in the periphery. And you can see just cut off that there was very high FDG uptake in the brain. And indeed, the brain is hungry for glucose and for its analog FDG, and is very amenable to study by FDG PET, if that's what you're interested in. It's not good for detecting tumor lesions in the brain, because these tumor lesions would hardly show up against the high background of the PET signal, physiological PET signal in the brain, but they show up very clearly against the cool background of normal muscle and tissues. So the brain has very high FDG uptake. Normal brain glucose burns glucose faster even than lymphoma lesions. And so it's been very useful for neuropsychiatry research to ignore the rest of the body that is interest to nuclear medicine doctors and focus only on the head. And this is a case study, or not a case study, a report in 3 patients with a particular variety of schizophrenia, catatonic schizophrenia, characterized by immobility and rigidity and various other aspects. And in the lower figure, we see their fluorodopa scans, which I guess showed the usual kind of tendency towards an increase. But uniquely or characteristically, in this subgroup of patients, they only had 3, because catatonic schizophrenia is rare these days. They all agreed in showing low metabolism, specifically in the temporal lobe on both sides. Maybe more on one side than the other, it's hard to say. But this was worth noticing, because it's not typical of schizophrenia in general, but only of catatonic schizophrenia. I don't know why that should be, but it shows that there's potential for physiological subtypes within categories of schizophrenia. In ordinary, that is to say, schizophrenia, the results are less clear, but here we see the results of a study from, it's an old study, but a very good one, I think, where they compared patients, fluorodeoxyglucose, that is glucose metabolism, in patients who had predominantly negative symptoms versus predominantly positive symptoms. A very key axis for defining different subcategories of the symptoms of schizophrenia. And what they found was very striking, that the negative symptom patients who were characterized by social withdrawal and behavioral changes, cognitive deficits, and so on, were hypometabolic, as shown by the blue. This is a statistical index comparing the 2 groups of brains, if you will, positive and negative symptom brains, measured with FDG PET. And interestingly, the hypometabolism was widespread over the cortex from the frontal, temporal, and occipital lobes on the right side. And once again, we see ourselves drawn to some idea that the brain asymmetry is contributing to the syndrome of schizophrenia. So the dopamine excess may be sometimes or often on the right-hand side, dominant hemisphere, and the hypometabolism specifically associated with negative symptoms is on the right-hand side. But you see the areas where the converse was true, where there was excess activity, were more symmetrically distributed left and right. So FDG PET gives a hint that there's some kind of lateralization of brain function in relation to the symptomatology of schizophrenia. Along kind of the same lines, the Oliver Howe's group in London, who are really the leaders in this kind of work right now, took fluorodopa scans, returning back to the dopamine pathway, in groups of young people who were prodromal for schizophrenia, who had the prodromal syndrome, which is highly predictive of conversion to schizophrenia in the near future, but did not yet meet the criteria. And they followed these patients after scanning them, and some of them converted. I forget, it was maybe half of them, and became psychotic. And then after the fact, they compared the fluorodopa scans of those who became ill and those who remained relatively healthy. And they found that the ones destined to become psychotic already had an excess of dopamine in the basal ganglia, symmetrically in this case, that seemed to predict or foreshadow that they would become ill. And this is, of course, a very interesting concept because it suggests that the dopamine defect precedes the actual illness and may be a risk factor in a sense. And it also implies that if one were bold, it might justify trials of antipsychotic medications in people before they become actually full-blown psychotic. So much for the dopamine system. It remains an active topic of research in molecular imaging of schizophrenia, but dopamine is not the only neurotransmitter. And the broad spectrum of tools for psychopharmacology research has been applied to different aspects of the brain of patients with schizophrenia, using a whole range of different radiopharmaceuticals, in addition to dopamine targeting radiopharmaceuticals. And one example of that is the muscarinic M1 receptor, which is a metabotropic acetylcholine receptor, or a class of 4 or 5 different receptors that are roughly similar, notably the M1 type receptors, which is the target of this notorious plant, the Jimson weed, as it's called popularly in English, that contains atropine and other atropine-like alkaloids that have medical use, but are also risky because their abuse, and some people, believe it or not, actually will eat these plants for, shall we say, recreational or adventure effects, at the risk of suffering a very severe acute drug-induced psychosis that has some, at least, overlap with schizophrenia. Now, the group of Therese van Amersfoort in Maastricht did a study using a ligand for M1 receptors in patients with schizophrenia. So they had no control group. It was all schizophrenia patients, but they measured the M1 muscarinic receptors in brain using SPECT, not PET, but it's the same principle, a radioactive molecule that targets a specific receptor, in this case, muscarinic receptors. And then they correlated the individual cognitive state of those patients with their individual muscarinic receptor binding. And they found various correlations, which here we see that they had about 25 subjects, which is nice, because you really need large data sets to discern these relationships because of the high individual variability. And within the population, there was a high correlation such that the more impaired cognitive performance, for example, in the delayed recognition task and other tasks, the lower the binding of the muscarinic receptor in hippocampus. And conversely, within those patients, those who had relatively preserved function had the higher levels. So it suggests that there's something to do with the individual degree of involvement of the muscarinic receptor system and the clinical manifestations in the negative symptom domain of that is more like cognition, that is kind of representative of the negative symptoms. So whereas dopamine results tend to correlate with the positive symptoms, as I probably should have mentioned, we see here that in this axis of muscarinic receptors, there's a kind of an inverse relationship. So this is relatively little studied. The paper by Therese Amersfoort was published a few years ago, and it follows upon a small body of work that fairly consistently shows that there's something amiss with the muscarinic receptor, but in schizophrenia. But on the scale of things, there are 100 dopamine papers for each muscarinic paper in PET and in molecular imaging. And I think that reflects a kind of a bias that we have, that it's easier to do dopamine studies, it's easier to get funding to do dopamine studies, but we may be missing things. We are missing things by not paying enough attention to other neurotransmitter systems that are also involved in schizophrenia, like the muscarinic receptor system. And now I turn to the final topic that I can cover in this talk in relation to molecular imaging of schizophrenia, with this somewhat ironic poster dating back to the 1920s, when there was or 30s, when there was a very active politically motivated campaign in the United States towards the prohibition of cannabis, which only became illegal about 100 years ago. I forget when exactly. And so there was a portrayal of it as being an exotic and dangerous, menacing thing that would lead to moral corruption and so on. I mean, that's what people thought, including the notorious, I would have to say, propaganda film Reefer Madness, which depicted cannabis as being a gateway to madness or psychosis. And drug crazed abandon. And that remains a kind of a controversial topic. I think everyone agrees that this was a propaganda film. And yet there is some association with, that's been shown in various studies in Sweden and elsewhere, that cannabis use, especially heavy cannabis use among young adolescents, seems to distinctly impart a risk for first manifestation of schizophrenia. Does that mean that it causes schizophrenia? That's a distinction that remains to be resolved. And we're sort of doing the experiment in my home country of Canada, where cannabis has recently become legal and in fact openly marketed a little bit too much. Everywhere you go, there are these cannabis shops here serving potheads, cannabis users since, I forget, which is humorous because the part of the phenomenology of cannabis intoxication is that people can't remember anything. So we're doing a social experiment. So far, as far as I know, there has been no big increase in the incidence of schizophrenia in Canada among young cannabis users, but we'll find out. Nonetheless, having put that topic aside, the cannabinoid receptor, especially the type 1 cannabinoid receptor in the brain, which is the site of action of THC, the constituent of cannabis, is another important target for research in schizophrenia, and one that's also relatively poorly developed, with only a smattering, a half dozen or so studies so far compared to the hundreds of the dopamine system. Now, this recent paper in JAMA Psychiatry from this a combination of Finland and London, which are two of the leaders in molecular imaging in schizophrenia. You will recognize the name Oliver House, and probably should remember Jarmo Hietala from Turku, who is doing great work on this. And they, for the first time, just a few years ago, did a PET study in patients with first episode psychosis using a cannabinoid receptor ligand. Now, I don't expect you to follow the chemistry, but I just want to convey an idea that making tracers for PET is not an easy matter. You have to use some pretty advanced chemistry. You need a cyclotron to make the F-fluorine 18, and you need special chemistry to integrate it into synthetic molecules to be used for proceeding from a precursor molecule here that is non-radioactive to the actual product, which has this chemical name. We prefer to call it FMPEP because it's much easier to pronounce. And it contains the fluorine 18 and has the chemical structure that makes it bind to cannabinol, the CBD1, the cannabinoid 1 type 1 receptor that's present in brain and is thought to be the main site of action of cannabis and related products. So summarizing their results in, I think it was 10 or 15 control subjects, we see that this cannabinoid type 1 receptor is very abundant throughout the cortex, maybe especially in the cingulate cortex, the limbic cortex. It's also present in the basal ganglia, the striatum, the anterior cingulate cortex, and maybe a little bit in the insula here. So it has an interesting distribution, but the remarkable thing that here we see the average of 10 subjects or so who were healthy controls, showing the distribution of the ligand binding to cannabinoid 1 receptors. And here in the lower part of the figure, we see a similar group of patients, none of whom were cannabis users. So there's no interaction here. This is the state of being untreated and with a diagnosis of schizophrenia, and having a very significant reduction in the abundance of cannabinoid receptors. So roughly speaking, too much dopamine, not enough cannabinoid receptors. We don't have scans in the same individuals with both ligands, but that would be something very important to do in the future. For example, it's possible that maybe there are multiple classes of schizophrenia, some with high dopamine, some with low FDG uptake, some with abnormal cannabinoid receptors. And we just don't yet know how to categorize them. They went further, like Van Amersfoort, they compared the results with cognitive scores, well, first with the PANSS score, and they showed the more severely affected individuals within their population, it looks like 15 cases, the more reduced the binding was. So in the hippocampus. So here's a pretty compelling correlation. The converse correlation was found with cognitive performance using a sort of a temporal lobe hippocampal task, an executive function task, digital symbol coding, and the more severely affected with the subjects with the less intact cognitive performance also were the same people who had low binding in, not in the hippocampus, but in the anterior cingulate, which fits with the executive function role. So they were able to tease out associations between how severely the cannabinoid system was affected with particular manifestations in positive symptoms and in a negative symptom of cognition. And that, I guess, is a new thing, because previously we had markers that were either for positive symptoms or negative symptoms. And here we have a marker that seems to relate with positive symptoms in one region and negative symptoms in the other region. So it might be that cannabinoid receptors will prove to be the unifying factor. So I conclude my talk, which is a very brief survey of a huge literature, to show that PET and SPECT imaging studies have implicated, among other things, dopamine receptors, muscarinic receptors, and endogenous cannabinoid systems in the pathogenesis of schizophrenia. However, no single marker is pathognomonic, my favorite word, but one I still can't pronounce, for schizophrenia, because all of them show extensive overlap, such that in a population there is an abnormality, but within individuals, they may fall within the normal or the abnormal range. So it's not diagnostic. So I conclude by suggesting that brain imaging, molecular brain imaging, captures only specific elements of a multi-dimensional space that might be represented by dopamine, glutamate still, muscarinic receptors, endogenous cannabinoid ligands, at least. And we don't know how large that list is, but it may take decades of continuing research with different tracers to kind of crystallize this into a correct and complete model that may establish what I would call a molecular typology of schizophrenia, which I think will prove to be not one. I'm sure we all agree that it's probably many overlapping syndromes, some of which may be dopamine driven, some of which may be more driven by other factors. So I think this is the task for molecular imaging for the next couple of decades. And that brings me to the end of my talk, and I thank you for your attention.
Dear Professor Paul Cumming, thank you very much for a very interesting lecture. And it seems to me that we're really moving to the whole course on neuroimaging in psychiatry. So it's your second lecture on molecular imaging in schizophrenia, and we would be happy also to know about MRI, fMRI studies in schizophrenia, and perhaps we can move also to the topic of neuroimaging in depression, and to work out the module on neuroimaging in neuropsychiatric disorders, which you might lead within this web library course. Thank you.
I'd be happy to. And I would add bipolar disorder, which I think is not talked about enough in this literature.
Absolutely, and bipolar disorder, and we can talk on neurological conditions as well, and as far as we can extend, and you will be, I hope, available for these lectures recording for us. Thank you very much for this collaboration. Thank you very much for this impressive lecture, thought-provoking, and you really inspire us to move into this topic and thinking about research which might be organized by our center in future. So thank you very much once again, dear Professor Paul Cumming. So, and hope to see you soon.
Thank you.
Bye-bye.